Hey everyone, this is Jyothika. Welcome back to my channel. Today we'll be talking about apoptosis. So we all know apoptosis is a small topic, and we know that apoptosis is programmed cell death. Apart from the definition, we have a lot more to study on apoptosis. We'll look into that. Now, so the topic today we are going to discuss is apoptosis, and the contents I'll be covering. We'll talk about the definition. We'll talk about the morphological feature. We'll talk about the mediators. We'll talk about the activation of mediators. So we'll discuss one by one. We'll talk about the definition. Definition is very simple: programmed cell death, energy-dependent programmed cell death. So this apoptosis occurs in a small group of cells or individual cells. That is all about the definition. Now we'll talk about the morphology. So when the cell is under apoptosis, in histological slide, we'll see certain features of apoptosis cell. So we'll learn about, so we'll talk about those features now. Normally, this is a normal cell. Now this is under apoptosis. For the first feature we'll see is the cell shrinks. The cell shrinks and the nucleus also shrinks, and this cells undergo fragmentation, and this fragmentized cell is taken up by the macrophage. This is taken up by the macrophage, and the important point is this apoptosis is not followed by any inflammation. That is the highest point you should know. So this is the morphological features, and in histological slide, the apoptized cell appears more eosinophil. It appears more eosinophilic, that is more pinkish. It appears more pinkish. So that is all about the morphology. Now let's go on to the most important content, that is mediators. How this apoptosis is mediated? This apoptosis is basically mediated by caspases, enzyme caspases. These enzymes are present in the cytosol. These are present in cytoplasm of the cell. Every cytoplasm of the cell, these caspases are present, and apoptosis is mediated by this caspases. This caspases in turn activates two more enzymes, that is endonucleases and proteases. This caspases in turn activates endonucleases and proteases. Now what does this endonucleases and proteases do? This endonucleases, we know, this will cause DNA to fragment. It will destroy DNA. And this protease destroy the cytoskeleton of the cell. So caspases, it activates endonucleases and proteases. Then this endonucleases fragments DNA, this proteases fragments cytoskeleton of cell. This is about the mediator. Now we'll talk about the activation of mediators. This activation of mediator, it is caspases only. Now how this caspases is activated? This caspases is activated in three different ways. One is intrinsic pathway, other is extrinsic pathway, third is CD8 T cell mediated pathway. So we'll talk about the intrinsic pathway now. Now this intrinsic pathway means there's some factors that are responsible intrinsically that are responsible for the caspases to activate. What are the factors? Those factors are like some cell injury or any DNA mutation that is occurring in the cell, inside the cell, that causes activation of the cell. Most importantly, there's one factor that is very important, and that is responsible for the activation of caspases. There is a BCL2 molecule in the cytoplasm. This is a mitochondria. This has cytochrome C. So the mitochondria has a cytochrome C, and BCL2 is present in cytoplasm. What is the connection between BCL2 and cytochrome C now? So this BCL2 will not allow the mitochondria to leak the cytoplasm, to leak the cytochrome C outside the cytoplasm. Because the cytochrome C has the ability to activate enzymes, many enzymes that are unnecessary for the cell and that is responsible for the cell death. So in order to avoid that, this BCL2 protects this mitochondria to not to leak out the cytochrome C. When the cell is under stress, when the cell is injured or the DNA is mutated, this BCL2 is inactivated. This BCL2 is inactivated. Now the cytochrome C is leaked outside the cytoplasm. Is leaked into the cytoplasm, and this causes the activation of caspases. This in turn causes the activation of caspases. That is how intrinsic mitochondrial pathway. This is how intrinsic mitochondrial pathway takes place. Now we'll talk about this extrinsic pathway. In extrinsic pathway, there is some factor outside the cell that is causing the cell to activate caspases. Are you getting? Like for example, I'll give you a small example. We all have T cells which are produced from bone marrow and matured in thymus. T cell, when this T cell go to thymus, they have to undergo two selections. For the T cell to survive in the blood, they have to undergo two selection in the thymus. That is, the maturation takes place in thymus. No, for the T cells to survive, the functional T cells are to be selected by the thymus, and they are released into the blood. So in thymus we have two selection process. One is positive selection and one is negative selection. In this positive selection, what does positive selection do? In this positive selection, T cell, the thymus will ask the T cell, it will check the T cell that whether it is capable of binding into the MHC class 1 or it is able to identify the self antigen. So if the T cell has the ability, then it undergoes negative selection. In this negative selection, it will again ask T cell, it will again check the T cells whether whether the T cell is able to bind to self antigen very strongly or just bind to the self antigen normally. So when this T cell has the ability to bind to the self antigen or MHC class 1 very strongly, then these T cells undergo rejection. This T cell undergo rejection. Why? Because in order to avoid autoimmunity or in order to avoid any graft rejections, this undergo negative selection. So why am I talking about this example? An extrinsic pathway, there's some receptor binding onto the cell and this causes activation of caspases and undergo apoptosis. So what in this process, there is Fas ligand that binds to Fas receptor. This is example, Fas ligand binds to Fas receptor. This is present in T cells. This is present on T cells, and this causes activation of caspases, and this undergo apoptosis. So this is how extrinsic pathway undergoes. So we talk about CD8 T cell mediated pathway. In this CD8 T cell mediated pathway, when the cell is stressed or is under stress or it is influenced, the CD8 T cell produce perforins. This perforins this will initiate pores. This makes pores on the cell membrane of the cell. This causes pores on the cell membrane of the cell. Then again, the CD8 T cell again it activates granzymes. This will activate granzymes. This granzymes will pass into the pores and will enter the cytoplasm of the cell. These granzymes enter the cytoplasm of the cell, and this will activate caspases and undergo apoptosis. So these are the three mechanisms on how this mediator. What is the mediator? Caspase. Caspases is the mediator. On how this caspases is activated once the cell is stressed. How this caspases is mediated, and this caspase, once the caspase is mediated, apoptosis takes place. That is all about the apoptosis. Thank you for watching.